I think everybody's probably getting pretty familiar with OCB and what it is at this point, but in case we have any newcomers today, I like to do this at the beginning for our virtual sessions. Here you just see the summer workshop website. Again, the green text is really where I want to go here. I really encourage you to have little regional node gatherings wherever you are, and interact with your colleagues. I think that connection is especially important right now. So I encourage you to do that. If you have any questions about the schedule or you're having trouble registering for the Zoom, obviously you're not if you're here, but if you have colleagues who want to join, feel free to encourage them. Everybody gets their own individual Zoom link, so please don't use other people's Zoom links, because then you come in as them, and then we have 2 or 3 people on the Zoom with the same name, and we're really trying to button things down and keep things secure so we don't have any weird incidents happening during our workshop. So OCB, really a big science collective working at the interface of climate, biogeochemistry, and marine life, working our way throughout the entire food web, but a lot of emphasis on the lower trophic levels. We have these large green icons here, which really encompass the scientific breadth of OCB. Most of what we do is within these green icons. And when we have this annual flagship summer workshop gathering, we really aim to kind of cross-cut and have some representation from all of those icons within our plenary sessions, because this meeting is really about bringing the whole community together and having the interdisciplinary dialogue that really launches a lot of great things science-wise. You can find all our information below. Website, e-newsletter, which is a monthly e-newsletter, comes to your inbox. Email address that comes to all 3 of us. The OCB project office is myself, Heather Benway, May Mahegan, and Mary Zawoski. Sure, most of you are familiar with us by now. We have a very active Blue Sky channel that has gotten a lot more follows recently. And we have a YouTube channel where we post all the recordings of our workshops and activities. The OCB project office would not be here without the sustained support of NSF and NASA, as well as our host, Woods Hole Oceanographic Institution. So big shout out to all of them and all of their efforts to help keep us whole right now. Just a couple quick announcements, because this is literally hot off the press. SOCAT 2025 just dropped today. So I wanted to put a shout out. All the info will be in e-news, and I also just put it out on Blue Sky, but I just wanted to let you all know that it's out and you can go and get that. SOCAT is the Surface Ocean CO2 Atlas. This is one of those ground roots community activities that, you know, doing it on a shoestring, but it's one of the most critical products this community produces. And a lot of you on this meeting this week have probably contributed to this at one point or another. A couple of other really exciting updates. EXOIS, which is, we've been talking a lot about marine carbon dioxide removal throughout the week. EXOIS is a collective of scientists focused on exploring ocean iron solutions, and the EXOIS program office, also based here at Woods Hole, is hiring, and it's a remote position, and it's only for the summer. So if you want to have the coolest summer job ever, I put the link down there. I recommend you check it out. It's a great team. I've worked with them, all of them, and they're fantastic to work with. Also, the UN Ocean Conference this week, a new MCDR report just dropped that Scott shared with me. So I wanted to put that out there as well. Both of these have been put out on our Blue Sky feed, so you can find them there. And with that, I want to get done so we can move on to science. So Faye, I'm going to let you take it away.
Right. Thank you so much, Heather.
Thank you.
I will just give a brief introduction to our last section of the session. So hi, everyone, and I'm sorry for the West Coast folks. It's very early for you. But today we will move downstream into the coastal ecosystem in our rivers to coast section. Yesterday, we started upstream in the watershed and heard fantastic talks exploring how weathering, erosion, degradation in the critical zone shape the generation, transport, and transformation of inorganic and organic materials at the headwaters. We then moved downstream to estuaries and wetlands, where speakers examined carbon cycling and fluxes across the land-ocean interface, with a focus on wetland transitions and their representation in budgets and models. Today, we'll continue that journey further downstream to the coastal ocean, where we will explore how coastal ecosystems generate, transform, and export dissolved organic matter, alkalinity, across dynamic nearshore environments. We'll begin with an overview talk by John, who will highlight long-term ecological research from 25 years of the Florida coastal Everglades lighter, a foundational data set for understanding coastal change and biogeochemical cycling. Next, we've shifted Gloria's talk from yesterday's wetland section to today to accommodate the different time zones. Her topic on alkalinity generation in blue carbon ecosystems fits just perfectly with today's focus on coastal and nearshore processes. We will then shift to the fate of dissolved organic matter in the coastal ocean. Cedric will present on advances and challenges in satellite remote sensing of dissolved organic matter stocks and transformations. Annie hopefully will be joining us. She just let us know she's not feeling well today, so I'm sorry to hear that. But if she could join later, she will talk about molecular level insights using high resolution techniques to trace the composition and fate of terrestrial DOM in Arctic systems. And after those plenary talks and a short bio break, we'll return for 3 lightning talks: dissolved organic carbon production by California kelp forests. Chance will be talking about that. And then Tahir, sorry about I butchered your name, microbials' role in ocean carbon cycling. And lastly, Shaily will talk about hurricane impacts on sedimentary silica cycling in the subtropical delta. Each speaker will present for 5 minutes to quickly highlight emerging research topics in coastal regions. We will close the plenary portion with 2 talks focusing on inorganic carbon fluxes and ocean acidification. So we covered both organic previously, and now we move on to inorganic carbon. Xinyu will present on anthropogenic carbon accumulation along the US East Coast. And on the West Coast, Simone will talk about insights from CO2 observing programs along the West Coast. After these talks, we'll bring all the speakers back for panel discussion. We will then wrap up the session with a summary and closing remarks from session organizers, Libby and David. Just a few quick reminders. All speakers will use their full 15 or 20 minutes for presentations in our session because we have so many fantastic talks. We will hold audience questions for the panel, but please feel free to drop your questions in the chat throughout the session and keep the conversation going, just like what we've done earlier in the week. For our speakers, if you can, please maybe use your phone to keep track of the time, but I will also give you a reminder if there's like 1 minute left. Okay. With that, let's just get started. John, if you can share your screen.
Good morning, everybody. Thanks so much for being here, especially the West Coast folks and folks overseas. So I'm the lead principal investigator for the Florida Coastal Everglades Long-Term Ecological Research Program, which is that cool 25-year LTER icon there next to the OCB icon on my title slide. We've been measuring data in the Everglades National Park in partnership with the Park Service and many other state and federal agencies. This is funded through the National Science Foundation, and I'm excited to share with you some of the large-scale changes that we're observing through our continuous monitoring for the last 25 years. So we just had our 25th anniversary. May 1st, 2025 was our actually coincided with our annual meeting, our all science meeting that we have each year. And sorry that advanced. So anyway, just that we're a large organization of colleagues from academic and non-academic institutions. Mostly students and postdocs are leading a lot of the frontiers of the research that we do. And feel free to join us if you're ever in Miami in the spring for our annual meeting. So hopefully we'll have one again next year. We're seeing some big changes in the landscape that mirror some of the things that people have talked about. Ghost forests, our mangroves are starting to succumb to sea level rise stressors. I won't talk much about that, but we're seeing large areas of peat collapse in our brackish marshes. So that's what peat collapse looks like on the ground. These sawgrass, brackish and freshwater marshes, when they get salinated, the root structure dies back and they open up into open water. The Everglades restoration started off in the late 90s, early 2000s because of harmful algal blooms and seagrass die-offs in Florida Bay. And so we're still seeing some die-offs during periods of droughts. The emphasis that our research program looks at is presses and pulses of fresh and marine water and how that influences vegetated coastal ecosystems in terms of their ability to continue to sequester carbon and perform ecosystem functions and services. This is our recent paper that my student Liz Ortiz put together. And it looks at all the compositional concentration, biogeochemical process changes, how altered hydrology and water management, all these things on land and water influence the carbon concentrations and compositions that get exported to the coast. And so I'm not going to, this is a review paper that was recently published in Limnology and Oceanography Letters, but I'm using these icons to guide some of the overview of this talk. So hopefully it'll help organize a little bit easier, but check out that paper if you're interested. So here we are in the Everglades. So for those of you who may or may not be familiar, the Everglades is a very large mosaic of freshwater marshes, brackish marshes, mangrove forests that drain into estuaries, and then out into seagrass meadows. And Florida Bay, down here at the bottom, is the world's largest continuous seagrass meadow. Shark River Slough is our main drainage, which drains underneath Tamiami Trail, which is currently being lifted up as one of the major restoration efforts of Everglades restoration. Everglades restoration is the world's largest ever attempted ecosystem restoration, and we've been measuring changes ever since the onset of that restoration for the last 25 years. These blue arrows represent freshwater pulses that are moving in Shark River Slough. The green arrows represent marine pulses that are coming in from the Gulf of Mexico and in through Florida Bay. Taylor Slough is a smaller drainage that's closer to Miami, and it is a marl-dominated drainage, lower productivity, less tidally influenced, whereas Shark River Slough is highly tidally influenced and is predominantly peat. Here's some videos just from the field to give you a sense of the different types of habitats that we have. Freshwater marshes are of sawgrass and sawgrass ridges and open sloughs that are full of carbonate, periphyton, organic matter. And we have urban and organic, or urban and agricultural areas off to the east in metropolitan Miami. Where the fresh and marine water mix is our ecotones, and they're dominated by mixture of marsh and mangrove species and some hardwood species. And then we have our mangrove forests that are both scrub mangrove forests associated with Taylor Slough, which is more phosphorus limited compared to these large, highly productive mangrove forests associated with Shark River Slough, which is less phosphorus limited, fed by phosphorus from the Gulf of Mexico. Okay. So here's what's happening in the Everglades. Sea level is rising, and freshwater restoration is also increasing freshwater to downstream marshes and mangroves. So this figure here shows 8 different locations across the landscape. On the left-hand side is Shark River Slough, so marsh, ecotone, mangrove, and seagrass out into Florida Bay. And on the right-hand side is Taylor Slough, that smaller drainage, marsh, ecotone, mangrove, and seagrass out in Florida Bay. The blue is the long-term average. I'm showing you water levels for the last 23 years. They're all going up. They've increased more recently from 2016 onward, coinciding with a rise in both rate of sea level as well as an increase in restoration efforts pushing fresh water into Everglades National Park. What we're seeing is that we can detect some of these storm pulses. We're also seeing the normal seasonal pulses, but water levels are not getting as low as they used to. In some cases, that's good. In some cases, that's concerning. And the magnitude of the water level pulse is going down, even though the amplitude is going up. How is that affecting dissolved organic carbon and dissolved organic matter? I'm going to talk to you about this from a few different projects. This first project was run by my former student, Kenny Anderson, and we looked at these boundary wetlands along Tamiami Trail and Shark River Slough and along this canal called the Aerojet Canal that borders Taylor Slough to the east. And so the Taylor Slough or the Aerojet are going to be the marl in tan, and the peat are going to be in dark gray for Shark River Slough. What we see is that as surface water increases in Shark River Slough, the peat, it actually decreases dissolved organic carbon, which isn't unexpected and has been shown by Rudolph Jaffe and others. What's interesting is in the marl-dominated wetlands that are predominantly periphyton-producing wetlands, not peat wetlands, we see an increase in dissolved organic carbon because that water that's coming in is coming in from other peat wetlands. Interesting. So when we look at the fluorescence index, which is a relative scale of microbial high to terrestrial low, we see that not unexpectedly, where we're increasing dissolved organic carbon concentration, we're decreasing the microbial component, increasing the terrestrial component in the marl wetlands, and changing slightly the microbial component in the peat wetlands. Biotic index, which is the algal contribution or the freshness of the produced carbon, recently produced carbon, we're seeing that there's less biological carbon, fresh algal carbon being produced in the marl wetlands with increasing surface waters, and not much of a change in the peat drainages. And we're seeing that humic index, the humic nature of the organic matter is increasing in the marl wetlands with that incoming fresh water. My former student, Matt Smith, who's was also a postdoc and now works up at the Army Corps, he looked at long-term concentrations of dissolved organic carbon, which are the black line. It's the same black line in all 3 figures, and compared those to changes in fluorescence index, which is up at the top in yellow, the slope ratio in blue, and the SUVA 254 in red. And so what we're seeing here is that microbial contributions of DOM are increasing, especially after floods and storms. So these dashed lines are droughts and floods and Hurricane Irma in 2017. So it really picks up the microbial components of DOM. Also, we're seeing an increase in the slope ratio with sea level rise over time. It's going up, but it's also very pulse driven by storms. And SUVA is decreasing when we have droughts. I'm sorry, increasing when we have droughts, but decreasing when we have increases in flushing of DOC. So just to give you a sense of the range of the different types of ecosystems, I mentioned that we have marshes, mangroves, and marine seagrass. So the dissolved organic matter associated with those spans that range, and not surprisingly, in the marine seagrasses, we're predominantly rich or higher in Bix, lower in Hicks. But we do have marshes and mangroves that span a range of the humification index. Similarly, the biological index is mostly strong on the marine end in the seagrass meadows, less so as you move inland. So what we're seeing comparing this PCA is, there's 3 PCAs, but what's shown here on the top left are the 3 different ecosystem types: marsh, mangrove, and marine, color-coded from 2011 to 2017, and then on the right top, 2018 to 2022. So we looked at these 2 time periods because it coincided with a rapid increase in sea level that I mentioned earlier. And a couple of things to note here is that marshes and mangroves are beginning to cluster. They clustered normally prior, but they're more clustering in association with humic and aromatic components. So that's shown here on the bottom PCA, the vectors. But then also, interestingly, the marine clustering is actually going away. It's expanding. And so we believe that that's being driven by more of these marine algal and microbial components. So the slope ratio, these components 6 and 5, which I haven't mentioned, are part of a Parafac model that we're using to interpret these data. And then the Bix, the biological, and the FI, the microbial, that I mentioned earlier. So we're seeing changes in the composition of this dissolved organic matter across the landscape. Okay, so just to characterize this a little bit more, Kenny was curious how much of this was changing in the downstream mangroves. So we've got Shark River Slough at the top and Taylor Slough at the bottom. We've got 3 sites of mangroves of Shark and 2 sites of mangroves of Taylor. And basically, the downstream sites of Shark and the downstream sites of Taylor are seeing an increase in the biological index associated with saltwater coming in. So there's more marine signature of biologically produced carbon coming in. And then what's also interesting is that those mangroves that are also downstream are producing a lot of carbon, and that carbon is being pushed upstream to the upper reaches of the mangroves. So at SRS4 and TSPH6, we're seeing an increase in Hicks moving inward with that water coming in from the Gulf of Mexico and Florida Bay. Okay, so these next 4 slides are looking at cross wavelets of time series comparing water level and DOC concentrations. So we have these measured monthly for 20-some years, and then we have more than a decade worth of the dissolved organic matter composition. So we're going to look at biological index changes and Hicks index changes. And so what we're seeing here in the freshwater peat marshes, so this is Shark River Slough's really carbon-rich peats, we're seeing that restoration is diluting. So as water level increases, DOC concentration decreases. So they're in antiphase or out of phase. And we're seeing less of an algal, so less Bix, and slightly increasing Hicks. If we go to the Marl site that I mentioned is getting a lot more humic, we're seeing that increases in water level are increasing dissolved organic carbon concentrations because that water is mostly coming from peat-rich marshes upstream. And we're seeing that they're in phase, but there's not a change in the Bix and Hicks over a water level. So it's pretty standard throughout the year. It's not seasonal. If we go downstream into the the mangroves of Shark River, we're seeing that there's a lot of flushing of dissolved organic matter into those mangrove estuaries from the marsh. And so as water level increases, DOC concentrations increase, and the biological contribution goes down, but the humic component stays. And then lastly, in that scrub mangrove forest of Taylor Slough, we're seeing that as water levels are increasing, both from freshwater restoration and from sea level rise, we're getting an increase in dissolved organic carbon concentrations. We're seeing less of it from biological and more of it from humic. Okay, a few last slides. Chris Osborne, who's a collaborator on our project, has been looking at the pattern of the sources of carbon using stable isotopes of carbon for the dissolved organic carbon. And what we're noticing here from a 4-year time series from SRS-1, which is that peat marsh, and SRS-6, which is the high productivity mangroves downstream, is that there's a synchronization between the concentrations and the isotopic value of DOC, indicating that there's seasonal shifts in the sources associated with the hydrologic shifts. And what's important to note here is that as DOC concentrations go up, the fractionation also goes up, so it becomes more depleted. And then lastly, Derek Vaughn, who's at Utah State University, but was working with Pete Raymond at Yale, they've been characterizing the alkalinity and the DIC flux, as well as the DOC flux, in Shark River Slough, which is the main drainage I mentioned, and a nearby Harney River Slough. And so what they're seeing is through 2 years of wet and dry season data, that we're actually having similar seasonal magnitudes in both the flux of DIC and alkalinity, but it's moving in opposite directions. And so during the dry season, it's coming further in with more saltwater intrusion. During the wet season, it's pulsed out with more freshwater flows. The fact that alkalinity is slightly higher than the DIC flux suggests that we're still seeing a potential carbon sink. And we're seeing more of these fluxes of greenhouse gases in the wet than the dry as expected, and it seems to be stimulated by storms such as Hurricane Ian. So just to sum up, the fresh and marine water presses and pulses, so from restoration and sea level rise presses, storm and seasonal pulses, they're shifting across the landscape and changing the concentrations and composition of DOM. And we're seeing higher water depths are mobilizing DOM from both marshes and mangroves. So pushing that DOM out from the peat down into the estuary and in from the mangroves to the upper parts of the mangrove ecotone. And then algal and microbial DOM is increasing with saltwater intrusion and sea level rise, and that has implications for carbon storage and greenhouse gas emissions. And we're continuing to characterize these long-term lateral fluxes of DOC and DIC in partnership with the Blue Flux team from NASA and other agencies and institutions. So I just want to acknowledge that a lot of this work was built by Rudolph Jaffe and Mei-Lian Chen and Nagamitsu Mei, who really pioneered the parafact model in the Everglades in the early part of our long-term ecological research program. And Chris Osborne and I are shepherding it forward and hope to continue to build it as these water and carbon fluxes are changing across the Everglades landscape. And I just want to acknowledge the LTR Network, the National Science Foundation, Environmental Data Initiative, the Park Service, and the U.S. Geological Survey who made this work possible. Thank you.
Right. Thank you so much, John, for that great talk. Very interesting. So just a reminder to our participants, please drop any questions you want to ask John. And then we will move on to the next speaker, Gloria.
So I'm going to be talking about alkalinity generation in blue carbon ecosystems and whether it's an overlooked mechanism for climate change mitigation. Very briefly about me, I'm originally from Germany, did my degree in environmental science. Then I did my PhD in Australia, where I focused on alkalinity export from mangroves. And now I'm in Sweden the last couple of years as a postdoc and now researcher looking at all the different blue carbon systems and their carbon budgets. Very quick recap, what are blue carbon ecosystems? Blue carbon ecosystems are mangroves and salt marshes, which are intertidal, and seagrasses and macroalgae, which are usually subtidal. Globally, mangroves mostly occur in the tropics and subtropics, whereas salt marshes are usually more found in the temperate areas. And seagrasses and macroalgae you find all around the globe, but different species. Blue carbon ecosystems are very productive and they store a lot of carbon in their biomass and in their soil. During photosynthesis, they take up atmospheric CO2 and then they store it in their sediments. This figure shows a comparison to other ecosystems, and you can see compared to the terrestrial ecosystems, blue carbon ecosystems store a lot more carbon on an aerial basis than, for example, compared to tropical rainforests. And they store most of their carbon in their sediments. Here you can see the mitigation potential, how much CO2 we can store if we just conserve the existing blue carbon ecosystems. It's shown in blue, and the orange shows the potential, how much we can store even more when we restore blue carbon ecosystems to their original area or just keep expanding them. And this getting the numbers right is very important. We really need to know where the carbon is going in blue carbon ecosystems because they're also used in carbon credit schemes. Now a bit more about my research. Before we heard about organic carbon more, and now I'm going to be focusing more about the inorganic carbon part. In general, more than a decade ago, they found that in mangroves, there is a large unaccounted carbon sink. So we did know the net primary production and kind of knew the barrier rates in the sediment, the CO2 or the greenhouse gas fluxes, and the organic carbon lateral fluxes. But there was a very large unaccounted carbon sink. And of course, to really get the carbon credits right and to really know how good blue carbon ecosystems are for the climate, we need to know where the carbon is going. And so far, and even up to date, most of the research has focused on the organic carbon storage in sediments. But then it was shown, this was one of the first studies that showed that actually quite a large amount is exported as dissolved inorganic carbon. Even more sometimes than the carbon stored in the sediments, but this was one of the first studies, so we really need to look at the full picture. So dissolved inorganic carbon might be this unaccounted carbon sink. About inorganic carbon, it exists, or we usually measure two forms. One is dissolved inorganic carbon, DIC, which also includes CO2. And this CO2 can outgas back to the atmosphere. Then it's, of course, not a permanent carbon sink if that's produced, and it can also enhance coastal acidification. Another inorganic carbon form is alkalinity or carbonate alkalinity. And in contrast to CO2, it stays in the ocean for millennia, so it is a permanent carbon sink. And it can also enhance the buffering of coastal waters. DSC and alkalinity are both produced in mangrove sediments, usually during aerobic processes, but at different ratios. So usually sulfate reduction is a very important one in coastal ecosystems. If we have some eutrophication, denitrification can also play a role. Like in the Everglades, for example, we have a carbonate basin, then carbonate dissolution can also contribute to the alkalinity generation. Here you see an idealized profile, depth profile in the sediments, but often all of these processes can also occur kind of parallel in the same depth, especially when we have barrows You see in mangroves, we have tidal pumping. So alkalinity and dissolved inorganic carbon are produced in the sediments. And then the pore water is highly enriched. And during high tide, the water comes then in and infiltrates the sediment. And then during low tide, it flushes out all this highly enriched pore water. And crab burrows on the right side here, you see a crab burrow in mangroves, which can be very large, and they usually build fresh during each tidal cycle. So there's a really large tidal pumping, and you can see big peaks during low tide. The concentrations of alkalinity and DSC are usually much higher. This is a quick overview of 2 different methods that can be used to investigate alkalinity and DSC fluxes on an ecosystem scale level. There are other methods that look at smaller scales, but in our group, we usually do the ecosystem scale. And one is the Eulerian, where you measure concentrations over time. This is very good to do when you have a well-defined creek, for example, and then you measure the water flow and the concentration, and then you see what's going in and what's going out. On the bottom, you see it's more, if you have an estuary, for example, as Shark River, then you need to do a spatial survey, and usually you can look at conservative mixing, and then you combine this with some kind of tracer to really also look at the source, where is it coming from, to attribute it definitely to mangroves, for example, versus freshwater input. This is the semi-recent study I did with 25 co-authors. I reached out to many people around the globe, asking if they could send me their data sets from alkalinity and DSC in mangroves and salt marshes. Some of these data sets are quite old, like from the 90s, where I think Excel wasn't really used yet, and some are really recent and very detailed, very high resolution. And because we wanted to know how big might this alkalinity export be. This is the global map we came up with. You can see for mangroves, we have a bit more sites. There usually the sample size is quite small, so we have often only one tidal cycle over 30 hours, versus for salt marshes, there are maybe fewer sites, but they're usually very well studied. We have nice annual studies or really long-term studies as well. But of course, there are big gaps still on the map, like in Africa and some parts of like Indonesia, especially, where we still need a lot more data. But what we did see is that mangroves are actually alkalinity sources. Almost all of them do export alkalinity, only sometimes very minor imports, but usually there are big sources, and the same for salt marshes as well. And also the fluxes can be quite huge. This shows my updated carbon budget for mangroves and salt marshes. You can see that that the DSC outwelling is the largest part of the carbon budget, usually almost like twice as high as the carbon burial. Right now, as I said earlier, the burial is still where the most research focus is on today, but DSC is twice as much, so I would say really it's an undercounted carbon sink. But of course, only if this DSC is outwelled in the form of alkalinity, and it usually is. Like the biggest part of DSC export is alkalinity. But you see there's still something unaccounted. We still have to fill this gap. I have to say that here I used medians, but if we use averages, we would have a closed, nice budget, and it's there is no unaccounted sink, but I think it's more honest to use medians because we have some very high outliers also for DSC outwelling. And now the next step would be to really look at what is still this unaccounted sink. So because if it's CO2 outgassing, which some people assume it is, then maybe they're not so amazing carbon sinks after all. But if maybe we do more alkalinity and DSC outwelling measurements, and it's actually that, then yes, that would be amazing, and they are even better than we thought. But there's still a lot of insecurity right now. I showed you this graph at the beginning about the mitigation potential, which we have if we conserve and restore blue carbon ecosystems. And this is just based on sediment and biomass. But if we now also account for alkalinity generation, which is twice as high as the biomass or the sediment, it looks actually like this. So yes, alkalinity generation in blue carbon ecosystems is an unaccounted carbon sink. For mangroves and marshes, we have some data, still not enough. But for the subtidal systems like seagrasses and macroalgae, we still don't know. And this is something my current research is focusing on, to get some numbers for seagrasses and macroalgae as well. Then I want to say thanks to the team who was all involved in this research. And if you're interested to know more, you're more than welcome to reach out to me.
All right. Thank you so much, Gloria. If participants, you have any questions, please type in the chat. Yeah, thank you. So our next speaker is Cedric. If you could please share your screen.
All right, thank you everyone. Thank you for the opportunity to present. So with this presentation, I hope to kind of give you my perspective on where I think we stand in terms of being able to do remote sensing of dissolved organic matter, salts, fluxes, transformations, really in the coastal zone. So I'll talk a little bit about the advances, challenges. So I'm pushing to the choir here, but there's lots of good reasons to study dissolved organic matter cycling. Right? This organic matter fulfills very important, multiple important biogeochemical and ecological functions throughout the land-ocean continuum. It's an important, so it's a large reservoir of carbon, a major source of reduced carbon and nitrogen throughout the land aquatic system. It's an important vector of carbon that can be exported to the deep ocean. This is also kind of vector for the production of refractory organic carbon through the microbial carbon pump, and its sequestration of organic carbon into the deep ocean. There's DOM also as a component of the DOM is colored, so significant optical properties. Which means it's an important regulator, one of the major regulator of the in-water light field, both in the ocean and also kind of the coastal and inland waters. Primarily kind of in the UV and blue part of the spectrum. Because it absorbs a lot of UV radiation, it's also a very important precursor for photochemical reactions, again, the productions of important compounds, climate relevant compounds. It's also a vector of contaminants and micronutrients. DOM kind of bound to some of these important kind of, for example, metals, and can serve as a vector for it. And even though it's more relevant here for freshwater system inland water, it's also a very important parameter to understand for be able to kind of measure for looking at drinking water, and it can also affect kind of the appearance of water bodies. And of course, these important functions, as can be strongly influenced by climate change, kind of leading to changes in the dynamics of DOM. So there's a lot of reasons to kind of trying to understand and monitor the dynamics of DOM, especially kind of in these estuarine and coastal system that kind of we're interested in here. This at this kind of interface of the ocean of land. It's this very dynamic, very strong dynamics of dissolved organic matter. So these are represent, in these systems in wetlands, in the coastal wetlands and the coastal ocean. DOM kind of represents kind of moderate but significant pools of DOM, right? Especially in comparison to the much larger pool of carbon that is present in the open ocean. But it's a much more heterogeneous and much more dynamic systems, right? Where you have influence from the land, influence from the ocean, currents, tides. And it's also kind of the location where you have multiple source and different types of this organic matter interacting. Well, autochthonous DOM, heterogeneous DOM, can also have anthropogenic inputs as well as atmospheric disposition. So very kind of important hotspots of DOM cycling. So this is kind of environment where tools like remote sensing that can facilitate observations on large scales and continuous observation can be particularly useful. And I guess we all know already that DOM to some degree is amenable to remote sensing, primarily through the detection of the optical properties of the part of the DOM that is optically active. Right, so with the for this presentation, really my objective here is to discuss a little bit about the amenability of DOM to remote sensing and really how this might be different depending on the types of environment, but kind of focusing on our estuaries and coastal system. I want to talk a little bit. I'm going to kind of review some of the recent applications, some recent, some less recent, but kind of see kind of what's been done. And I'm going to be a little bit biased towards kind of my own work, and then also kind of present some other works. And then I will briefly talk a little bit what's next, what's exciting next. So I guess one of the main kind of take-home message I want to present here is that the amenability of DOM to remote sensing is very variable across the land-ocean continuum. And by amenability, I really mean the feasibility of being able to monitor DOM and calculation of fluxes and transformations, and with sufficient accuracy that it can be useful. And so that if you look here, kind of in this at the top here, right, it's kind of amenability meter. Okay, we can see this kind of variations across the land-ocean continuum. Right? It's very conceptual here. It's kind of very general, but it's kind of generally true with of course with some case-by-case changes. But you can see kind of amenability being relatively poor in the waters, kind of moderate in open ocean, and being a little better kind of the type of environment here that we're interested in, such as coastal wetlands and the coastal ocean. And really the kind of this amenability is largely driven on the factor of from three primary depends on two primary factors. One is kind of the strength of the robustness of the CDOM as a proxy for the rest of the DOM pool. How good of an indicator it is for some of the kind of variables that we're interested in, and how representative of the full pool it is. And this kind of can be variable across these different environments. In general, relatively good, although can be kind of a little bit local. But can be generally good across, from inland waters to the coastal ocean. A larger decoupling of those two in the open ocean. I'm not gonna talk much about the open ocean today, but in general, this relationship, can be variable, but generally relatively good, relatively robust in this system. The other important consideration is really the optical complexity of the environment, and by this I mean the optical complexity of the water themselves. So, is there the presence of, sediment, for example, or other phytoplankton that can really strongly interfere with the detection of it. But also, we're talking about the complexity of the atmosphere that can really influence the accuracy of the reflectance measurement that we can use to determine CDOM. And that's something, again, that is increasing as we go towards inland, and also the presence of adjacency effects. So this optic complexity of the water body, of the atmosphere, and adjacency generally increase as you move inland. So it's just better in the open ocean, being much more complicated in inland waters, and kind of moderate in between. And then finally, the last consideration is really the spatial and temporal scales, and whether they match kind of the type of dynamics that we see in these systems. So obviously, a lot of the satellites that are currently kind of present, with ocean color satellites with resolutions of several hundred meters to a kilometer, generally kind of appropriate for open ocean and coastal system, becoming inappropriate as you start to move much more near shore, right? And then there's other types of satellites that have a high, finer spatial resolution but with coarser or coarser, more infrequent measurements that may not be able to capture quite adequately the dynamics in these nearshore system, even though the spatial resolution is better. So this, considering all these kind of different aspects, okay, you can see kind of drives the range of amenability in these different systems. So in general, the system that we see for coastal and for the coastal ocean and coastal wetland are generally relatively good based on the satellites that we see, but there's still some limitations, especially near shore, as maybe some of the resolution can be limiting. So we have a wide range of sensors that are, some of them are not in operation anymore, but we have a wide range of sensors that are available for this kind of measurements across these systems, and with sensors having better spectral and good coarser and good temporal resolution, but coarser spatial resolution. Other sensor like Sentinel-2. Or OLI, having finer spatial resolution, but being more limited in terms of spectral resolution and temporal coverage. And of course, we have PACE coming up with appropriate primarily for the ocean and having hyperspectral characteristics that will open kind of new applications. All right, so going back a little bit to what's been done in the field. So one of kind of the important kind of improvement that has been made in more recent years is kind of the use of the spectral slope coefficient to 75 to 95 as an indicator of the ratio between CDOM and DOC. As I mentioned, the important consideration is how good CDOM is representative of the greater DOM pool and primarily the DOC concentration in this case. It's been shown in kind of Gulf of Mexico across different seasons where the relationship can be, between DOC and CDOM can be variable. The spectral slope is a very good indicator of the ratio. That's something that's been confirmed in other systems like Chesapeake Bay or the Mid-Atlantic Bight. Again, providing kind of a handle on being able to constrain that ratio that can vary on seasonal basis or across the gradient. And this kind of has helped, be able to kind of a more generalized kind of algorithm for retrieving DOC concentration. In this case, across large estuaries of the U.S. So this is kind of using kind of the MERIS at 300-meter resolution. So some of these kind of resolution, useful for larger estuaries. As you start to go into closer to land, you would benefit from finer spatial resolution. And so the new kind of suite of sensors from Sentinel and Landsat 8 have better radiometric resolution. At the 10 to 30 meter spatial resolution, kind of facilitates the detection of DOC concentration in a lot of the system and start to kind of get a glimpse at the dynamics in this system. One of kind of the limitation, however, kind of these sensors that they only kind of able to provide observation every few days. Here's an example of where the combination of Landsat 8 and Sentinel-2A and 2B kind of provide 3 images within 8 days, which is pretty much as good as it gets. So now this can become limiting, when you start to look at the dynamics, the kind of tidal oscillation could be limiting potentially to get a picture of the tidal dynamics in the system. But what we showed kind of more of in a recent study is that now kind of the record, the multi-year record of some of these sensors, for example, in this case, just of the Sentinel-2, can be sufficient to start to get a better understanding of these dynamics. Or kind of get a better, more complete picture of these dynamics, even though you cannot, acquire data every hour for these tidal systems. And here's an example for some work we did at the Plum Island estuary, where the conditions are kind of favorable to get good simple algorithms and good DOC retrievals. You can see here along all the gray points along those lines for kind of different discharge level, different level of productivity of the marsh, and different tidal conditions. It's like over a 7-year period or so, there's enough observation to capture really the wide range of conditions that are typically encountered in the system. So, and this is something we've been able here to leverage to be able to develop a very simple model, statistical model, where we can predict the DOC concentration throughout the estuary, or at least along the estuary, based on simple variables such as the EVI, so the vegetation index over the salt marsh, the river discharge, and the water level, to be able to kind of reproduce here the DOC concentration. Here's kind of a validation, an independent validation of these estimates we made from the model that was kind of trained using the remote sensing measurements against independent measurements. So again, showing that with long enough of a record, even though the temporal resolution is not necessarily appropriate for these systems, you can still kind of better understand the dynamics in this system. All right, so going more into the coastal waters, I'd like to illustrate here a bit of work from Antonio Manino, where they've been using a large data set, in situ data set, to develop robust algorithm and facilitate the detection of DOC across the entire shelf. Be able to combine this to over multi-years using models to calculate stocks of DOC. Combining this with the ROMS kind of base model, they've been able to quantify kind of fluxes of DOC across those different interfaces and across the shelf, right? To get at this kind of essentially integrated kind of fluxes, and be able to kind of address budgets of DOC in this coastal environment. So this is kind of one of the nicer application combination of a model and remote sensing data. So these coastal systems, so environments where you have a strong mixture of different types of DOM. This is kind of going back some work I did as part of my PhD, where we demonstrated that the use, essentially, the use of the empirical algorithm of the spectral slope coefficient and its calibration using the lignin biomarker, we've been able to derive kind of a simple algorithm to actually look at more the heterogeneous component of the DOC. So you can see here the concentration is much lower. It's because it's only representing the heterogeneous component. And we'll be able to reuse this and apply this to the time series to actually demonstrate kind of the pulse nature of the export of heterogeneous system in this environment. So these were strictly looking at concentration to look at variability, but you could see how you could combine this also with model to get more specific export of heterogeneous DOC across the shelf, instead of just the full pool of DOC that was done for the Manino paper. I'm going to skip on this because I'm running a little late, but we've been here just briefly. We've been able to use demonstrate that you could use some hyperspectral data from a airborne sensor to trace the effluent, leveraging the fluorescence properties, the effects of the fluorescence of effluent on the reflectance. Briefly to mention, we've also been able to leverage kind of remote sensing to facilitate the quantification of photo bleaching across waters. We've put a lot of work and be able to kind of develop a model that can help us predict the apparent quantum yield metrics, or the efficiency essentially of solar radiation to lead to the degradation of the CDOM properties. And you can see how across what we discovered is really across the land ocean continuum, you have this organic matter really tends to bleach differently. We show that this metric was largely predictable from the spectral slope, the water temperature, and the measure of the extent of solar exposure, which we've been able to, these are kind of variables you can retrieve or calculate from using kind of remote sensing data. And so be able to use this information to get a kind of a global picture of the variability of the apparent quantum yield, and implementing this into a kind of a global kind of photo photo bleaching model be able to calculate kind of the first global rates of photo bleaching rates and the first climatology and get a sense of the relative contribution of the importance of photo bleaching as a cycling mechanism for CDOM in the surface water. So similar work can be done, for looking at photochemical production of climate-relevant species such as carbon monoxide, methane, or glyoxal, which is an important precursor for secondary radical formation. Secondary aerosol formation. Sorry. So briefly to conclude, where do we stand and what's exciting coming up? I think we're at the dawn of kind of hyperspectral satellite observation. Especially we're particularly interested in be able to use kind of hyperspectral data and the use of reflectance, UV reflectance, which this is part of the spectrum where the optical properties of the OM kind of is particularly dominant, and we kind of hope to improve estimates of DOM from this part of spectrum, as long as we can get good reflectance from space. So in that context, the use of PACE is kind of particularly exciting for the coastal ocean. I haven't had the chance myself yet to play a little bit with this data, but hoping to do that in the near future. And of course, Glimmer, providing kind of these high temporal and more high spatial resolution in coastal waters, and potentially SPG. We'll see where that lands. Okay, thank you. And if you want a little more information about this, I can point you out to this review paper. Thank you.
All right. Thank you so much, Cedric, and thank you to our previous speakers, John and Gloria. Yeah, so please feel free to check out her paper looking at the fate of dissolved organic matter from Yukon River in the Arctic Ocean. Yeah, so maybe we can take a couple minutes to answer some questions using Annie's time.
Okay, yeah, so it's the question was, what is the ocean color proxy? I'm estimating to use a UV spectral slope. So usually this is based on this is purely empirical. This is based on a multiple linear regression. Okay, using multiple bands from the blue and the red, so trying to leverage the entire spectrum. This worked remarkably well. I know it's fully empirical, but it's worked remarkably well across a wide range of systems. And so,
Yes, I have a question for like I'm a modeler, so but I really appreciate all this data. I think it's like one of the foundations for developing models. Like for example, John, the system you are looking at seems like this saltwater intrusion tidal cycle seems to be very important. Are there like any advances in like sensors measurement techniques to help you like capture this like full tidal cycle? I know like sometimes maybe if there's no like in situ sensor, you may need to stay over the night to collect sample every hour, or yeah. So just broadly, if there's any advance in that area.
Like the $1 million question, Faye. I appreciate you asking it. The short answer is we need sensor instrumentation, like FDOM sensors. I mean, that they're not the only answer to capturing this, or CO2 sensors. It's tricky going into a wilderness area in a national park to get permitted. We do sampling. We have some of the samples collected continuously with autosamplers, but for dissolved organic matter and DOC, we collect them monthly, and we know that we're missing a lot. So the work that the Blue Flux team from NASA and Yale that I showed some of the data on DIC and alkalinity, those were samples where they were able to get out on a boat and do overnight sampling, as well as do, they're actually down there now doing 2-week sampling, so going out every other day. So we have some of those concentrated efforts, but across a multi-decadal time series, we don't. And so we are talking about how to pivot in our, one of the challenges with long-term data is how do you transition methods? How do you transition what you sample, where and when? I don't have a great answer for you other than that we know we are missing and we know we need to collect in a different way. So we're trying to prioritize that. We have been doing it more at in our ecotone sites where we can kind of at least capture the fresh and the marine signals, but even then it's not been continued for as long as we would like.
Okay, I see.
Yeah.
Thank you. Thank you so much.
Sure.
Yeah. Seems like there's another question for Cedric on photo bleaching from
Yes. Yeah, so it's Mara's question. Yeah, how quantitatively important is photo bleaching in the open ocean for DOC degradation compared with other losses of DOC? So I think here I want to really distinguish the photo degradation of DOC, photo mineralization, which maybe is what you're referring to, versus photo bleaching. And I'm sorry if I didn't explain this in the talk, but photo bleaching really refers to the loss of CDOM optical properties upon solar exposure. So this is not photo degradation, it's not the photo mineralization. Here we quantify the photo bleaching, that loss of CDOM. And so what we estimated kind of globally is that in the surface mix layer, solar exposure photo bleaching cycles or goes through the cycles through the whole stock of CDOM 6 times per year. Right, so which means there's other kind of mechanism kind of resupply. So it's just calculating kind of the based on the rates of loss. In terms, so this is significant, for it's a significant regulator of the optical properties in the surface water. Now, if we're talking about photodegradation, I think it's much smaller. This is much smaller, and this difference between the photomineralization versus photobleaching is also probably one of the important mechanisms that leads to that decoupling between the CDOM and DOC kind of a relationship in the open ocean.
Okay. Yeah, welcome back, everyone. Maybe now we can move on to our lightning talks.
Hey, thank you. Hello. I'm guessing everyone can hear me well. Hi, so my name is Chance English. I'm a recent PhD graduate at the University of California, Santa Barbara. I'm currently working with Craig Carlson and Dave Siegel. So today I'll be talking about some of my dissertation work looking at dynamics of dissolved organic carbon production by California giant kelp forests. So motivation, so kelps dominate temperate coastlines around the globe, and they're proposed to sequester large amounts of carbon, largely in the form of dissolved organic carbon. However, studies on the magnitude of DOC released by macroalgae show this very large range in the fraction of net primary production that's partitioned into DOC. It's from anywhere from 1 to like 76%. So understanding the controls on DOC production by kelp is really important for integrating them into coastal blue carbon budgets. And so to do this in this study, we wanted to measure the partitioning of net primary production into DOC by the canopy-forming giant kelp across these gradients in environmental conditions like light intensity, seasonal nutrient availability, and age-related physiological gradients. And so we did this by incubating giant kelp blades sampled from tagged cohorts across the natural lifespan of giant kelp fronds, and then we repeated this sampling from cohorts established in the nutrient deplete summer and nutrient replete spring at a Santa Barbara coastal LTR site in Mohawk Reef. And I'll note that I won't have time to discuss these results, but we also have data on the composition of the DOC released and the makeup of the kelp microbiome. And these seem to be a really important part of the story that we're presenting here, but I won't have time. But I'll have a series of QR links linking to these a couple of preprints and a paper that just came out on these with more detail. So as I mentioned, we conducted incubations from a tagged summer and spring cohort shown in those highlighted areas along the plot. So on this plot, we're seeing temperature. This is average daily temperature, Mohawk Reef, and the black line. And in the blue line, we're seeing average daily nitrate concentrations calculated from a temperature to nitrate relationship established for our study site. And so we can see in the summer, we captured this period of high stratification, low inorganic nitrogen availability throughout the summer cohort. We can see within each period, we sampled kelp across a gradient of physiological conditions. So the vertical dashed black line showing kelp in a mature physiological state, and the vertical dashed red line showing kelp sampled as in a senescent state. And we see when we get into the spring cohort, we capture this period of kelp being exposed to these high nitrate concentrations as a result of spring-driven upwelling, wind-driven upwelling in the spring. So we observed a strong response of both kelp physiological condition, so measured as the chlorophyll carbon proxy, and rates of net primary production in response to these environmental gradients and age. On the left, the chlorophyll carbon ratio is a proxy for kelp growth rate. We see large seasonal difference at the initial values, so higher in the spring makes sense. There's more inorganic nitrogen availability. But we see that in both cohorts in the spring and summer, there's this rapid nonlinear decline beginning at about 50 days, which is indicative of kelp senescence. And then on the right plot, we're looking at the relationship of net primary production to environmental gradients like light intensity and age. And so we see this predictable relationship with net primary production and light. But what's notable here is this linear decline in the maximal net primary production rates with age, which is this sort of telltale signature of a senescing autotrophic organism. And so when senescence is playing a really important role in the physiology and the net primary production, we wanted to integrate how does this affect the partitioning of DOC if we consider the entire lifespan of kelp. So we can go to the next slide. So if we look at the relationship between DOC production and net primary production in mature kelp, what we see on the left is that DOC production is a really small, relatively constant fraction of net primary production and responds to the same sort of environmental variables like light intensity. So for mature kelp, DOC is really only about 3% of net primary production. But on the right plot, what we see is if we observe kelp throughout its lifespan, there's this large decoupling between DOC production and net primary production as kelp ages beyond 50 days, so that senescence mark. And in many cases, DOC oxidation is actually greater than or equal to the simultaneous rates of net primary production, indicating this loss of previously fixed carbon. So we wanted to combine these incubations and physiological experiments to the sort of California-wide giant kelp area. And so in order to do this, we used a long-term Landsat-derived time series of giant kelp biomass. And so that biomass is shown in a plot on the right. And so we have monthly estimates of total standing canopy biomass throughout the state between the years 2001 and 2023. And then also because of the frequency of the Landsat sensors for our region, we're actually able to we were actually able to estimate daily changes in biomass of kelp canopy along this time series. And so we're able to sort of assign this standing biomass a physiological state, so either mature or senescent based on whether it's older or younger than that 50 days. And what we see is for biomass, there's this really large intra and interannual variability. So canopy biomass has this, any standing canopy biomass at any given time can range between 2 and 370 gigagrams of wet weight. But if we look at the physiological state, so this bottom plot, what we're looking at is that there's this pretty regular recurrent pattern in kelp senescence throughout the year across a large scale, the California-wide scale, which is lowest in the spring, so about a quarter of the biomass, and then peaking in the fall about 3 months after the peak in biomass in the summer, where almost 3/4 of the biomass is senescent. And so basically showing that senescent biomass is a really important component of kelp physiological state at a large scale. So you can go to the next slide. So when we incorporate our kelp senescence DOC production rates into an annual budget of DOC production by giant kelp using our estimates of standing biomass and kelp physiological condition for our region shown on the left. So on the left plot, we're seeing average standing biomass of giant kelp along the central and southern California coastline, bend at 500-meter latitudinal bends. We see on the right, if we include or do not include senescence, we see this large difference in the annual amount of DOC produced by giant kelp. And so actually when we include senescence, we find it's responsible for almost 3/4 of the DOC produced by kelp. And then on top of that, there's this large interannual variability we can see, and that's largely a function of this huge variability in standing biomass. So I think really this is showing that one of the critical pieces to integrating kelp into blue carbon estimates is having estimates of how much standing biomass there is, just because it's such a transient population. and really have an understanding of its physiology in order to sort of integrate these sort of previously overlooked mechanisms of DOC production by kelp. So you can go ahead, go to the next slide. So there's a lot more to say, obviously. So I linked to a couple of studies that have just come out of our group. So on the left is a preprint soon to come out within the next couple of weeks. On the left, and then in the middle is a study of the kelp microbiome. And I think really these large pulses of DOC we're seeing in senescent kelp are likely this result of these growth of these bacteria that are solubilizing tissue and solubilizing that into resulting in these plumes of DOC. And then on the right, I think of interest is not just how much DOC gets released, but how much is actually bioavailable and how much is recalcitrant. And we have a study with a different macroalgal species showing that photooxidation is really an important component of the turnover of sort of more recalcitrant dissolved organic carbon released by brown seaweeds. And with that, I will end my talk. Thank you. And thanks to my funding agencies, Department of Energy, RPE, and support and long-term data from the Santa Barbara Coastal LTER.
Okay, Heather, there are a lot of animations in my presentation, so sorry. So I'd like to talk to you, oh, hang on, let me just start. I'd like to talk to you today about a project that we had looking at hurricane impacts on silica diagenesis. And this was a project with Jeffrey Krause, along with the Extreme Silica crew, pictured here in the bottom corner. Jeff has a great way of coming up with project titles. So he was really channeling the X Games for this. And a big thank you to NSF. The project resulted in several dissertation chapters, 3 undergraduate projects, and it's the basis for 2 more chapters for my students. So we're going to be focusing on this land-ocean interface next, and looking at what a hurricane is going to do to this system. So if you, next, yes. So I was really motivated by the ABT framework we heard about on Monday. So I'm going to try it. You can give me feedback, so go ahead. Next. So diatoms, responsible for more than 15% of global oxygen production, are the base of healthy coastal marine food webs, and silica is an essential nutrient for their cell function. Next. But we don't know how extreme physical disturbances that stir up the seabed, like hurricanes, will impact their biogeochemical cycle. Next. Therefore, we investigated the impacts of Hurricane Ida in the Mississippi Delta before and after, next, to quantify silica regeneration or bioavailability from the benthos to the water column. Okay, next. And Hurricane Ida was a Category 4 hurricane. So you can see where it made landfall, right where we sampled for these 3 stations. So we sampled about 2 weeks before the hurricane made landfall, and then got funding to sample 4 more times that year. So we're in less than 50 meters of water depth, and the hurricane likely stirred up 15 to 20 centimeters of the seabed, which we know by excess lead-210 activities. Next. So when you're in the open ocean, typically, this is in sediment, so pore fluid, dissolved silica profiles usually asymptote after about 20 or 30 centimeters. So this would be a typical open ocean profiles. Next. And so this is really consistent with this canonical model of silica diagenesis, where the diatoms or biogenic silica that reaches the seabed, most of it dissolves and diffuses back into the water column where it's bioavailable. Next. After hurricane in coastal systems, these are the pore water profiles that we observed. So the red line is the profile before the storm at one of our stations. The orange is the profile about 4 months later, and the blue is a profile 1 year later of DSI. Now, these are really unusual in that they have a maxima, and then they decrease with depth, which indicates that there's a reaction that's consuming dissolved silica in pore fluid. And we can investigate these reactions by looking at stable isotope, silicon isotope profiles. And you may not be used to seeing stable silicon isotope profiles. So the solid red triangles are the signatures of BSI. And if BSI was the only thing that was dissolving in pore water, the open triangles are the dissolved fraction, they should track right one on top of the other. And so the typical range of delta silicon 30 values on surface earth reservoirs range from -2 to +2. And so these are some of the most positive, like the highest dissolved delta silicon 30 values that we've ever observed, to my knowledge. There's almost a 1 to 2, almost 3 per mil difference between the solid phase and the dissolved phase. So this is inconsistent with this canonical model of just dissolution and refluxing back into the water column. And we observed this in all of our sampling seasons in December and in August. And so what that indicates is that you need this revised model of silica diagenesis where the DSI that's present in pore fluid reacts, which is what is leading to this fractionation in your stable silicon isotope ratios. And two of my students, Indigo and Claire, are going to be working on quantifying the kinetics of these reactions in other deltaic systems and in the Mississippi. Now, secondary and tertiary motivation for this is that when BSI undergoes this diagenetic alteration in this yellow box up here in this revised model, it produces CO2. So this is a concern for MCDR technologies. You're looking at alkalinity enhancement technologies, and also the reactions that control atmospheric CO2 over geologic timescales. So the main takeaways from this project is that we observed that the benthic fluxes of DSI to the water column appear to be elevated 1 year after a hurricane. Now, this is inconsistent with responses from carbon, which may respond, say, in a 1 or 2 month time frame. And hurricanes may alleviate regional nutrient stress in coastal systems. But at the same time, when you have this alleviated benthic flux of DSI, maybe from sediment to pore water, at the same time, you also have diagenetic alteration of BSI, which is rapid. But to really resolve all of these issues, we need a regional mass balance by monitoring a whole suite of inputs and outputs like currents, rivers, and groundwater. So with that, I will end my talk. Thank you again to NSF.
Right, thank you so much, Shaily. Yeah, I saw many great conversations in the chat, so please keep it rolling. Our next speaker is Xinyu, who will talk about anthropogenic carbon accumulation.
All right, thank you so much for having me here. And before I start my talk, I would like to thanks for the session organizers. And this is a really nice session. And I'm very happy to see like the people who work on the river and the estuary and the coastal ocean. I really enjoyed all the talks during the past 1.5 days, and also the OCB organizers for having such an amazing summer workshop every year. So today I'm going to talk about the anthropogenic carbon along the US East Coast. My original title also involves some ocean acidification effects, but then I realized the audience of this session probably more interested in the carbon transport, so I just dropped it. So the atmospheric CO2 concentration has increased due to human activities such as fossil fuel burning, land exchange, and deforestation. And this figure on the right is a record of the atmospheric CO2 increasing from the 1960s. So the ocean is a very large carbon reservoir. The dissolved inorganic carbon in the ocean can be approximately divided into the natural component and the anthropogenic component. And the figure on the right, this is a depth profile of the DIC and also the natural component and anthropogenic component in the ocean. For the natural component, it's more depleted in the upper ocean and becomes the concentration becomes higher with the depths. And the anthropogenic component is the opposite, is more enriched in the upper ocean. And the important thing I want to mention here is the anthropogenic component is a very small signal and cannot be directly measured. And what we can measure is only the total DIC, a combination of the natural and anthropogenic component. However, the anthropogenic carbon can be estimated through the repeated hydrographic courses. And here is a plan back to the 1990s and all the way to the current. And the map in the middle shows the major transects of the repeated hydrography where we have constant and high quality dissolving organic carbon measurements in the ocean so that we can estimate the anthropogenic carbon. And with that, we know pretty well about the anthropogenic carbon content in the open ocean via the repeated hydrographic transects. However, we don't know much about the coastal ocean. And in this kind of global analysis, the coastal ocean is either neglected or just do a very simple linear extrapolation from the open ocean to the coastal ocean. So the knowledge gap here is we don't know much about the coastal ocean anthropogenic carbon distributions, the inventory, and also the mechanisms. And as we all know, coastal ocean is a very complex system, and it involves the estuary wetlands and also continental shelf part. And also there's a lot of dynamic process going on. So just a linear extrapolation is not really enough to let us understand more about the coastal ocean anthropogenic carbon. However, estimate the anthropogenic carbon in the coastal ocean can be very challenging. Here is a very simple figure I draw, and to show on the axis is the time and the y-axis is the DIC. Let's assume that's our measurements. And the issue here is that we usually have very short observation records, and also the natural variation of DIC is very large. So that means the anthropogenic carbon signal is very small. So in order to solve this, I think we have 3 key points we need to make. And the first one is that we need the high quality and very consistent data. And the second point is we need to long-term observations. And also we need a very effective methodologies to estimate anthropogenic carbon in the coastal ocean. The first component, high quality data. So the NOAA Ocean Acidification Program have supported lots of coastal ocean acidification cruises that provide a lot of high quality carbon measurements. And here I'm going to use the East Coast Ocean Acidification Cruise as an example. And this was a cruise I attended twice during my PhD work. So the ECOA cruises covers the regions of the most of the US East Coast and also some Canadian coastal ocean as well. And it's repeated every 3 to 4 years since 2015. And the figure on the left is a map of the ECOA cruise track, and the East Coast cruise can be divided into 3 major parts. On the left is the South Atlantic Bight from the Florida Strait to the North Carolina. And also in the middle is the Mid-Atlantic Bight, and on the north is the Gulf and Main region. And this kind of cruise provide very high quality data of carbonate measurements to detect the climate level change. And also, if the program continues, it provides very precious long-term records for the analysis. Okay, then let's sum up what we have so far. And we have some long-term DIC observations. And the ECOA cruise I just introduced before, it covers the 3 regions. And before that, we have the GOMAC cruise. The GOMAC cruise on the East Coast part mainly covers the South Atlantic Bight region and also Mid-Atlantic Bight region. And even before that, back in 1996, we have an ocean margin program, and that is only limited in the Mid-Atlantic Bight region and has high quality anthropogenic carbon measurements as well. So in the following talk, if I'm talking about the whole East Coast, in the South Atlantic Bight to the Mid-Atlantic Bight, I'm using the data from the GOMAC cruise and the ECOA cruise, and I'm talking about the time period from 2007 to 2022. And if I'm only zoom in the Mid-Atlantic Bight region, and I'm talking about like a longer time scale from 1996 to 2022, just for clearance. So this is a depth profile of Massachusetts transect in the Mid-Atlantic Bight. And we have the longest records here and dating back from 1996 to 2022. And as you can see, that the DIC concentration, it seems like increasing, but not really. I think because the ocean is dynamic, the water mass can be moving, and also probably there is some measurement uncertainties going on, and it makes detect anthropogenic carbon directly and seeing the DIC inventory concentration growth very difficult. That's why we need a method. We have everything set up, and then we need a a good method to estimate the coastal anthropogenic carbon. Here I'm going to introduce my own method, RICA. It's a regional empirical regression-based anthropogenic carbon estimations. And I'm not going to get into all the details of this method, but I would like you to know the key points of this is we put in the hydrographic parameters we measured, such as temperature, salinity, oxygen, to do a regression with DIC, and then we can calculate the anthropogenic carbon. And this method is based on the open ocean anthropogenic carbon estimation algorithms, and I did some regional modifications that's suitable for dynamic coastal oceans. So we want to validate this method, and we validate using the ocean system simulation experiments. And the key idea is that, assuming we have a digital world, the data from the digital world are from the recap2 model output, and we know everything in the digital world. We know the temperature, salinity, oxygen of all 40 years and everywhere by one multiply 1 grade cell. And also we know the anthropogenic carbon in this system through different simulation rounds. So what we do is we pretend we go on cruise in the digital world, and we follow exactly the same track as we did in the real world. And then I apply my algorithms to this synthetic data and to get the anthropogenic carbon estimations. And I compare the estimations with the known true value, so I can get a sense of the uncertainty of this method. So I test this method in all major US coasts, the Gulf Alaska, West Coast, and upwelling region, and the East Coast is West Boundary region, and the Gulf of Mexico, which is a river-dominant region. And here's our result. And the color bar here shows the differences of my estimations subtract the model true values. It's a bias. So once you see the color of each tiles becomes more pale, that means the bias is smaller. So as we can see here on the West Coast and also the East Coast, the model performs better than the more complex systems such as Gulf of Alaska and the Gulf of Mexico. But overall, this method can capture the anthropogenic carbon in the coastal very pretty good, and I'm decided to use this to the real data. And the next, I'm going to use the East Coast intensively as an example to see the anthropogenic carbon distributions and what can learn from that. So here, it shows the anthropogenic carbon distributions along the US East Coast between 2007 and 2022. On the northern part is the Mid-Atlantic Bight, and the southern part is the South Atlantic Bight region. The first findings we found is that the South Atlantic Bight anthropogenic carbon accumulations is higher than the Mid-Atlantic Bight. As you can see, the color is deeper. And the second takeaway is that the anthropogenic carbon accumulation depth profile have a sandwich structure. The sandwich structure, is high in the upper layer water, which is Gulf Stream water, and then becomes low again in the Antarctic intermediate water, which in the middle is also a very oxygen depleted water with very high AOU, and it becomes high again in the North Atlantic deep water. Probably it's not very clear to show in this map, but I'm going to show a temperature salinity plot, which can show the water mass more clear and to demonstrate this point furthermore. And the third point is the anthropogenic carbon accumulation increase from near shore to the offshore. And this phenomenon is especially clear in the Mid-Atlantic Bight. As you can see, the nearshore part has a lighter color, which means less anthropogenic carbon accumulation during this period, and with it increasing. Okay, I'm just going to dive slightly deep into the subregions and also across different time, because as I described before, we have lots of cruise, and we can just do a comparison, not only between 2007 and 2022, but also we can do a comparison between different time periods. In the South Atlantic Bight, the the temperature and salinity plot can show the different water mass very clear. On the top is Gulf Stream water with high anthropogenic carbon concentration, and then move to the Antarctic intermediate water with very low anthropogenic carbon, and North Atlantic deep water with high again. And this is mainly due to the water mass age and also water properties. It shows the different buffer capacity to accumulate anthropogenic carbon. And also for the North Atlantic deep water, it has a water age about 20 years. So what we really see of the anthropogenic carbon accumulations between 2007 to 2022 is actually a reflection of atmospheric CO2 increase 20 years ago. That means between 1987 to 2002. And for the Antarctic intermediate water, as the name, it travels all the way from Antarctic Ocean to the to my observation area. So the water age is very long. At that time, we don't have very much anthropogenic carbon accumulations. So that's why we see such a low signal here. And it's the same. We're just going to move from the South Atlantic bight and to the Mid-Atlantic bight. The main feature here in the Mid-Atlantic bight is we see an anthropogenic carbon increase from nearshore to offshore. And this pattern is consistent from 2007 to 2022, and also other time period as well. The nearshore low salinity part have less anthropogenic carbon accumulations, and offshore Gulf Stream water has more. And now I'm going to zoom into the Mid-Atlantic bight with the longest records, and I plot the anthropogenic carbon distributions, especially on the shelf, and to see the distributions as well, and also what we can learn from that in terms of land-ocean transportations. So in the Chesapeake Bay transect, Delaware Bay transect, New Jersey transect, and also Massachusetts transects, that's the 4 major transects in the Mid-Atlantic bight region, we can see the consistent pattern of the low anthropogenic carbon in the low salinity part and high anthropogenic carbon in that, in the offshore part. What does this tell us? So the coastal ocean is a link between land and the ocean. And what we used to think is a very simple river pipeline model. That means what is in the river and all dumped into the ocean. But with knowledge advanced, and we know that's not easy. I know this figure is a little bit overwhelming, but I want you guys to zoom in the land to ocean part. And the continental shelf box is what my research area focused on. And the nearshore to the offshore gradients we see is the export of the estuary and tidal wetlands to the continental shelf waters. And the red numbers here is the anthropogenic carbon change of total, including the carbon, inorganic carbon, also organic carbon part. And this is the number from the Rainier Nature paper.
Xinyu, can you please wrap up? We are closing to that time. Yeah.
Yes. So with the Mid-Atlantic bight region, I did a very similar budget analysis. The yellow number is my analysis. Because of the low concentration we have, we conclude that there is a little anthropogenic carbon export from the estuary and wetlands. And on the continental shelf part, we have very active export of anthropogenic carbon, the uptake from the coastal oceans. Well, that's actually my last slides. So I'd like to thanks for my PhD advisor, Dr. Wei-Jing Tsai, and also my postdoc mentor, Dr. Brandon Cotter, on supporting this work, and also the Ecola course PIs, and also all of my other collaborators. And with that, I will leave the take-home message here. And if you're interested in more about the anthropogenic carbon budget, you can scan the QR code on the red top, and you will find more details in the paper. And that's it. Thank you.
Right. Thank you, Xinyu, for the great talk. Yeah, now we can move on to our final plenary talk for the session. Simone, if you could please share your screen, turn on your camera.
thank you to the organizers of the meeting and this session for inviting me to be part of this today. It's been a number of years that since I last attended OCB, so it's really nice to see so many familiar faces, even if just virtually. So today, my talk is going to be more focused on the ocean acidification and ecosystem condition piece of this session's theme. And I'm going to try to give you a tour through kind of what we've learned about the influence of rivers, respiration, ocean acidification, and extreme events on ecosystem conditions over a broad swath of the Pacific coast. So I'm showing you on this slide some of my key thought partners over the years, and certainly many other people have contributed to the work that you'll see today. And I want to acknowledge our funding sources. Most of them are NOAA sources, but we also are fortunate to receive funding from the Washington State Legislature. And then just briefly at the end, I'll describe a little bit about some of the work that parallels what I'll talk mostly about today in Alaskan waters that I've become more involved with since Jessica Cross left PMEL. So briefly, the Pacific coast, what I'll talk about mostly focuses on this temperate coastal rainforest region here. We've been doing these observations since 2006. There are two major oceanographic current systems here, both upwelling and downwelling. We have complex geomorphology, including fjords as far south as Puget Sound. We've got two major river systems in this area, the Columbia and the Fraser, and many small mountainous rivers. There's also major river system further south. And throughout this region, we have a variety of river inputs from glacial fed to rain or snow fed, with more river discharge in the north. Of course, the nature of the hydrology feeding these rivers can affect the timing of discharge, as well as the biogeochemical composition of these waters, which I'm not going to focus on today, but it's very important for the downstream effects that I'll talk about today. I also want to briefly tell you about the Salish Sea observations that we've made since 2008. The Salish Sea comprises the Strait of Georgia, the Strait of Juan de Fuca, and Puget Sound, which is the focus of a lot of my work. You can see our sampling stations in the interior of Puget Sound in the inlet here. And this ecosystem also has very diverse habitats and a decade of seasonal cruises. So at PMEL, we do, as Xinyu and others have referred to very nicely, a lot of open ocean observations in the North Pacific, as well as coastal observations. We also have more time series, but then you can see in this IPAC OA screenshot here, there are a lot of other observations that we're fortunate to have on the Pacific coast, including land-based and near-shore systems and gliders as well. And if you zoom in kind of more to the coastal view, you can see the spatial footprint of the West Coast OA cruises on the left, and again, the Washington Ocean Acidification Center cruises on the right. This is WCOA and WOAC, somewhat confusingly. So I wanted to start by talking about a few of the key insights that we've gained from WCOA cruises since the first one in 2007. On the left, you see the figure that launched 1,000 studies. This is Richard Feely's first publication about OA in the California current system, showing the depth of the Aragonite saturation horizon. At the time, it was a surprise to see it extending to the surface in places. But you can see that we see that sort of pattern on pretty much all of these cruises. This is the plot on the right from the 2021 cruise. And our next cruise is scheduled for next summer. But since those sort of early simple views of the status of ocean acidification on the Pacific coast, we've gotten more into estimating anthropogenic carbon. Xinyu gave a great introduction to that. So I'll just say that in these maps of surface sea anth concentrations through the cruise years, you can see, well, you can't see because I'm only showing you the surface, that it's highest at the surface as she reflected on. But we see also somewhat higher values across depth and possibly higher rates of subsurface increase to the south in the California current system. In addition to leading that part of our analyses on this, Brendan Carter also estimated the biological DIC component in subsurface waters where respiration plays a strong role. And you can see in these maps through the years at 50 meters on the top and 100 in the bottom, that if anything, we have a tendency towards higher respiratory CO2 in the Northern California current system. And there's very strong spatial and interannual variability across cruises. We've also learned about rates of acidification in this coastal system compared to the open ocean. In a paper by Mar Arroyo a few years ago, they compared North Pacific ghost ship line here with a coastal transect. And these plots on the right show the change since the pre-industrial of aragonite saturation state in the top plots and PCO2 in the bottom plots. And basically the point I want to communicate about this is that you can see with the warmer colors and the plots on the right that the overall change since the pre-industrial has been larger in the coastal water masses because of differences in buffering capacity relative to the open ocean. And rivers play a part in that, of course. We also have had a large body of work looking at the dissolution impacts on pteropod shells, mostly led by Nina Bednarczyk. And both in the open ocean and coastal ocean of the North Pacific, we have relatively large percentages of aragonite, of pteropods with severe dissolution. She's developed those as water quality indicators as well for coastal states. But increasingly, we're looking at more direct fishery management species of focus, such as a Dungeness crab. Dungeness crab are consistently the top valued West Coast commercial fishery and have acidification sensitivity across life stages. The plots here are my analysis of time series mooring data from Olympic Coast National Marine Sanctuary, shown in this map at the lower left. And basically using data from the West Coast OA cruises, Brendan Carter's CANTH estimation routines, and these time series data, we've been able to look at spatial and temporal variability across this suite of moorings. And you can see that as we go through time from the pre-industrial to the end of the century, which is based on Sam Sidlecki's ROMS model work, you can see an increasing presence of conditions that may be harmful to Dungeness crab on the Washington shelf through that time. Okay, this is a very busy slide. I'm not going to go into a lot of the details, but we also are working more on multiple stressors. All of these are fundamentally carbon cycle problems, temperature increase over the long term, marine heat waves, seasonal hypoxia, which we also have on the shelf, and then also changing hydrology and increasing harmful algal blooms. So these plots are from the WOAC Puget Sound cruises, and the magenta lines show Dungeness crab specific sensitivity thresholds. The plots are set up with surface data on the right from April, July, and September, and deep water, on the left, and deeper water on the right. And you can see that we have a lot of conditions that hit both sides of those thresholds for multiple different stressors related to Dungeness crab. Okay, very briefly, this is the same kind of plot, but looking at FCO2 through Puget Sound across basins. And here, most of our values are above atmospheric values, which is indicated by the yellow lines. Across basins, we see more variability and higher FCO2 values in stratified areas than better mixed basins. But also, kind of here's again where river and land influence comes in. We see stronger depth gradients, and we can see really severe conditions in deep water in those river-influenced, very productive basins. Okay, so looking at that river-influenced basin, I want to show you briefly how extreme events have played out in the Salish Sea And to do so, I'm comparing cruises from 2018, a more normal year, with the peak heat wave year of 2015. In Hood Canal, normally conditions are quite severe, both in July and September. But you can see in 2015 and 2016, conditions actually improved by this September cruise, and that was due to anomalous circulation related to the heat wave. See a similar story for aragonite saturation with the same comparison plots here. The differences between these months may appear to be slightly less because aragonite saturation is already so low here. And I don't expect you to be able to see all the details of this plot. The arrows just point to the traces from coastal moorings that I was speaking about earlier, showing that during the heat wave years, conditions didn't get either as intense or as long lasting on the coasts. Okay, now another kind of extreme event that we observed through the Salish Sea ecosystem cruises is a CO2 storm. And I'm calling it that after Waldbusser and Salisbury's paper on carbonate weather a few years ago. And so these plots show aragonite on top with 2017 now compared to 2018. And on the bottom, same thing for FCO2. But here, where we saw the really anomalous conditions was in the main basin, which is normally not as acidified. And there in 2017, the conditions were 0.3 to 0.4 lower for aragonite saturation and 200 to over 600 higher for FCO2 throughout the water column. So this was something we'd never seen before. And I'll note, we do a lot of work estimating carbonate system parameters from temperature, salinity, and oxygen. And we would have missed this because oxygen was not particularly low at that time. We saw the same sort of pattern at stations in the Strait of Juan de Fuca a month later. So it did seem to be fairly widespread. And ultimately, after thinking of a lot of different options, we concluded that these conditions were fundamentally driven by anomalously high river runoff in the spring of 2017 that set up long-lasting stratification and sustained bloom conditions, ultimately leading to a huge respiration signal in the water column. Okay, in terms of drivers of estuarine and coastal acidification on the West Coast, I've talked a bit about rivers in the past few slides, talked a bit about respiration in this paper on decadal oxygen decrease on the Pacific Northwest shelf. Jack Barth's data from 2021, including the Wacoa cruise, show these hotspots of respiration in areas with longer retention times. But you can also see really well the influence of the Columbia River input, which is within this circle here. And that's true also for acidification levels. So in this 10-year year-day mean look at acidification conditions in surface and bottom waters, so this is aragonite saturation, you can really see the influence of the Columbia River off of the mouth year-round in terms of lower saturation states, as you might expect. And when the plume is blown to the south during the upwelling season, you see lower saturation states to the south. And then in bottom waters, you see this correspondence of the very worst conditions that we see below 0.5, both north and south of the Columbia River mouth. And this is some really beautiful work that Kate Hewitt is currently writing up, so keep an eye out for that. Okay, calcite saturation. I don't know how much this community thinks about calcite undersaturation, but given the importance of Dungeness crab, we have thought about it more recently. We routinely see it in deep Hood Canal waters. This is 5 years of cruises. All the warm colors indicate undersaturation. We did observe it also in that CO2 storm in the main basin. Wiley Evans's team has observed it many years in the Strait of Georgia. And so, the point here is calcite undersaturation is actually more widespread and frequent than hypoxia. And this appears to also be true on the coast. And so I guess one thing I haven't really called out is that the rates of change for FCO2 and aragonite saturation differ in this very already acidified set of systems that we're looking at on the Pacific coast. And that's because fundamentally, again, of differences in buffering capacity across basins, depths, and time. And of course, buffering capacity is affected by river input, by biology, predominantly primary production and respiration. And upwelling could be changes in winds or water mass, et cetera. But in this part of this plot here, where the CO2 and carbonate ion content curves cross, you can expect to see CO2 change accelerate. So the increase in FCO2 accelerating once we're on the left side of that curve, and saturation state changes decelerating. We do see that in these ecosystems at the low pHs that we have in many parts of this region. This is just, again, a similar box plot showing you that in the Pacific Northwest, well, in Puget Sound, we see a lot of DIC to TA ratios that are close to or over 1, which puts us in this territory. Okay, I'm sorry I'm a little bit over. I will wrap it up in the next few slides. When we first started thinking about this, we thought, mostly about the patterns and processes in the ocean environment. The kind of human implications were a little bit more of an afterthought or an add-on, as you see in this figure. We were thrilled to have the first ocean acidification program funded regional vulnerability assessment, allowing us to really dig into tribal and commercial use, mostly tribal use of marine resources and their vulnerability to ocean acidification changes. And I also like the inclusion of oceanographic use of the ocean in this slide as well. Okay, I won't go into this. I just want to encourage you to look at this review led by Brian Hunt in LNO last year. In it, we get into the potential climate change effects on changing hydrology and coastal ocean biogeochemistry and other aspects of coastal oceanography in this region. And briefly, I promise to not do it justice, and I won't, but getting involved with similar work in Alaska has been really rewarding. We're trying to currently plan the next generation of OA observations in Alaska. And this is with Natalie Menacci and Darcy Dugan, along with OAP. They're starting to see similar impacts on commercially valuable crab populations in Alaska as well. And the observations and modeling work coming out of that region are being increasingly incorporated into ecosystem status reports and other management processes. Okay, I think I'll just leave this here for the brief moment while we change slides. I missed the pet happy hour, so here are mine enjoying some ice cream. Thank you.
Great. Thank you so much, Simone, for that great talk. Yeah, now it's time for the panel discussion. Our first speaker yesterday for the river section, Susan Anderson, wasn't here yesterday, but she pre-recorded talk. She was at DC for the congressional visit to advocate for federal funding, but she's here today. So if you have any questions, want to ask her during the panel discussion, please feel free to ask. And go check out the recording if you haven't. And if you want to reach out to her, I believe send an email will be fine too. Yeah. So do we have any questions for the speakers? I saw some great conversations in the chat there's a question for Simone from Alec in the chat.
Okay, Alec, thank you for the question. Interactions between OA and HABs, in fact, we have a project funded on this right now from OAP and I think National Ocean Service to look at harmful algal bloom species of concern to Salish Sea tribes and marine resources. And so that involves an experimental component as well as some data analysis. And I mean, the short answer is yes, there are a number of different ways that ocean acidification and the other stressors that I alluded to can affect HAB. So, it does seem like we're seeing, greater incidence of HABs and new occurrences and so forth in this region. And so yet another way that this becomes a great laboratory to look at multi-stressors in this changing ocean. And, that project I alluded to is kind of in middle stages, so I don't have specific results to share with you, but, feel free to reach out later and we can talk about that more. Thank you.
Right. Thank you. Colleen, could you please ask your question? Or you raised the hand.
Yes, I also have a question for Simone. So Dungeness crab, all crabs are modal, at least in the benthic stages. So I was wondering, what are the spatiotemporal scales where these stressors start to impact them?
Oh my gosh, do you have an hour, Colleen? It's so interesting.
I do.
And awesome. And I don't consider myself, an expert on that, but I did try to dig in a bit to understanding the situation more as part of various projects related to this. But yes, they move. Some of their life stages are kind of more near shore, like brooding, or water column based, like the larval and juvenile stages. And so it's incredibly complex to think about what the bottlenecks are. But in terms of their mobility during the benthic stages, it's fascinating because we see some years on the coast really terrible conditions, and then they'll have just an absolutely great fishery catch year. So some of the work that Samantha Sedlecki's been leading involves creating seasonal forecasts of ocean conditions, and that work includes co-developing information for tribal and state fishery managers. And so, we quarterly or semi-annually get updates on how the fisheries are going and so forth. And so I think we have yet to really get to the bottom of that. But the bad conditions tend to occur at times when the fisheries are the least active. And so, I don't know if the crabs are migrating more near shore where conditions are somewhat less bad, and therefore avoiding those conditions that may otherwise be harmful, and or potentially running into new food sources that then, contribute to successful fisheries. So I don't know, stay tuned. It's fascinating to see that that situation with seemingly great fishery results while we're also telling them conditions are very bad. And I guess it reminds me that yesterday was looking at some of Paul McElhenney's work, and it suggested that under acidified conditions, there may be more but smaller larvae. And I guess I would say that it occurred to me that in Alaska, I think they're seeing a similar thing with the salmon fishery, where there's like more individuals, but they're smaller. And so I guess I would just really go out on a limb and say, that may be where we are in the evolution of coastal and climate conditions, that, initially populations may be responding by having seemingly greater success in terms of numbers, but at the cost of perhaps having smaller individuals. So I don't know. There's a lot yet to be learned about that, but just some thoughts from the file. Thanks.
Thanks, Simone. It makes me excited to think about modeling the crab population there.
All right, thank you. There's another question for Simone. Maybe we can come back later, give you some break. For Gloria, there were two questions earlier. If you scroll back to the chat around like 10 o'clock, there were questions for you on alkalinity fluxes. One from Zach, one from Jennifer. Yeah, if you could talk about that.
I counted carbon sink in the carbon budget I made, and I have to clarify the carbon budget I made based on different review papers, so it was not site specific, but I, for example, had a barrier rate from the most recent review paper about barrier rates and so on for each of the carbon components, and of course that will probably also contribute to this not matching or lead to this unaccounted sink. But there were not, for all the alkalinity sites, there were not carbon barrier rates and greenhouse gas fluxes and so on. So it was not possible to do it for each of the sites, which would have been probably more accurate. And then also, yes, there is a very large variability at each site. I tried to build a global model based on tidal amplitudes, temperatures, and so on, but it did not work because the variability is higher than the global one. So I could not, it is probably also definitely a big factor. And another one is also the different methods. For example, for the Everglades, we did spatial and temporal measurements and compared the fluxes, and they were really different, even though it was from the same day. So I think overall, it's probably, yes, definitely a systematic issue, but still, I believe that we don't have enough sites to really be sure, and we still need to have more measurements before we can definitely say what really is the reason for this unaccounted sink.
Right. Thank you. Thank you so much, Gloria. There's a question for Xinyu in the chat.
Okay, I'll just directly to answer. And thanks, Shane, for your answer. So the Cape Hatteras is actually a very tricky transact to handle with. And the way I do this in my analysis is I separate them to the South Atlantic Bight and Mid-Atlantic Bight, right? And along the transects. So in those two different regions, I use different regression models. Because for the South Atlantic Bight, it's mainly influenced only by the Gulf Stream, and the Mid-Atlantic Bight, actually, we see more interacts of the terrestrial inputs and the offshore inputs. So the short answer for the Cape Hatteras transects, in my analysis, I consider it's more like the South Atlantic Bight region, and it's mainly influenced by the Gulf Stream. And I was not really able to see much of the interaction of the terrestrial input and the offshore inputs at that specific transect. And we were expect to see more gradients and changes in the Mid-Atlantic Bight.
Thank you, Xinyu. While we're on this topic, I have a question for you, Xinyu. That's very exciting to see this method. I remember you mentioned in like systems like Gulf of Mexico, Gulf of Alaska, it is not as good as like other regions. Why is that the case? Do you have a sense like how to improve the model? Can it be used further in like more complex systems like estuaries in those regions? Yeah.
So for the Gulf of Mexico, one key missing point in all models I've tried for the OSI experiments is they're missing the river inputs a lot. So that's the key struggle from the model perspective in the Gulf of Mexico. And for the Gulf of Alaska, the situation becomes more complicated because in those kind of simulations, we're expecting the ocean should more or less keep a steady state point, which means the temperature, we're not expecting the temperature, salinity, oxygen change a lot during the time period I'm observing. Otherwise, the anthropogenic carbon estimation method, it has a natural flaw in these algorithms. But for the Gulf of Alaska region, from all the model results, we see a very obvious temperature increasing and oxygen decreasing. And I think those two parts are the reason why the algorithms results doesn't really match the true results from the model. And from my personal perspective, I also don't have any clue of whether it's the algorithms or it's the model.
Yeah, great. Thank you. I saw Shaily mention that SGD may also be one reason for that. Yeah. Okay, cool. Right, Simone, do you want to answer the question from Zach earlier?
Oh, thanks. Sure. Zach, yeah, great question. And, you know, I can't speak definitively to that, but there's also the crab fishery in the Southern Hood Canal that's closed. And so, I mean, South Sound is shallow and tends to be the warmest. And so, and also the area where you'd have maybe the greatest concentration of anthropogenic nutrient inputs and so forth. So there's a lot of complexity there in terms of knowing for sure why that fishery is closed. I feel like I heard recently that temperature is kind of a leading thought, but don't quote me on that. And then I think the contrast between Hood Canal and South Sound opens up interesting opportunities for kind of looking at different impacts. And Larissa Diaz, who I believe is also on the call, just got a project funded to work with Puget Sound data, as well as some of the ecosystem managers around here to maybe look into things like that a bit more. So hopefully we'll hear from her in a future year about that and what we're learning.
Okay, cool. Thank you. Jen, do you want to ask a question?
Yeah, sure. I just, throughout the talks, they're fantastic. I've just kind of realized a theme of there's a lot of extremely complex processes happening and terminology. And, I feel like we can reach a broader audience if we can try and figure out how to maybe simplify these processes for people in other disciplines. And I was wondering if any of you have given that some thought. And I know you work across fisheries and ocean observations, probably a lot of physical oceanographers and whatnot. And I just feel like maybe there's a gap there and that I'd love your input on.
I mean, I'd say a few words. I don't want to dominate, but giving others a chance to think about it. I guess I'd say I have two thoughts on that. One is I always kind of resist the idea of simplification because I think we need to understand the complexity, but like that's, I guess, for us. And then, of course, figuring out how you communicate with your various audiences. You really have to do, I think, as Catherine Hayhoe has been a great proponent of, think about your audience and figure out where the common ground is to really tailor the message. And so, like for me today, I took a talk I gave to a much broader audience, non-carbon focused last week, and felt free to not include certain explanatory details, you know what I mean? So like, and still I'm sure it misses some when we do that, but yeah, just thinking deeply about what contributes to a particular conversation, what doesn't, is an ongoing process, I find.
I'll chime in too. We think about this quite a bit with the work in the Everglades in terms of communicating it to managers and policy for restoration. And so like water movement makes a lot more sense to people who can control the flow of water or movement of water into one location or another, or restoration is successful if in the mind of the manager if animals and plants are responding positively, but like carbon is hard for them to see. And so we try to emphasize that systems that are developing, that are accumulating carbon, are going to have long-term better benefits to the system, to both humans and nature, and that we want to retain carbon on the land more than we want to lose it to the atmosphere or to the ocean. But that's not always what we want, it's not always as easy as that. So trying to bundle a lot of complexity into an outcome is, I think, your question is a good one. And actually next week I'm heading to DC myself and trying to speak to an audience of whom I don't even really know who all is going to be there, but they're not going to be scientists, and talking about how we move forward to make better measurements that people can understand, that can be accessed more in real time, so that people seem to value, because people value things that they can access, whether or not they understand it, right? So a lot of these data are hard to understand, and we make them available in ways that most people who aren't using them don't understand how to find them. So we need to do a better job somehow. That's our challenge.
Right. Thank you, John and Simone. Any other speakers want to chime in?
If I could just add a few words to what John said, I think one thing I've really been edified to see over the last several years is an increasing focus on funding data product development through NOAA's ocean acidification program. So that allows us to kind of create useful things out of the observations that we make for people who may have interest and stake in understanding that information, but not necessarily the time or knowledge base to do it themselves, right? So a lot of cool partnerships have emerged from that.
Do you have any other questions? Right.
I have a question for Suzanne. Generally, I was wondering if you could, I remember your slide about this intrusion, the propagation of cracks through rocks and this water rock interaction and breaking of bonds. I was wondering if you could just speak a little bit more broadly to lithological controls on your solute fluxes.
Wait a minute, solute fluxes or breaking bonds in rocks?
Both, since they would be connected.
I mean, I talked about subcritical cracking and what happens that extends cracks there. And I don't know, I'm not a rock mechanicist, don't do rock mechanics, but you can imagine that the bond strength actually matters for the propagation of the cracks. And so, yes, there's a difference from one type of mineral and rock to another, I think, in what that strength looks like. In terms of solutes, of course, it matters a lot what mineral you're weathering. The rate at which a mineral will react depends on what its composition is, and carbonates are very much more susceptible to weathering processes than silicates, than quartz, and and then there's the whole variation between feldspars and micas and quartz. All the different silicate minerals have different reactivities to them. So yes, lithology matters a lot. And then there's the interactions that I tried to discuss, that it depends on what the weathering environment is. And if you have, for instance, disseminated pyrite in a rock, you're going to be producing, likely, if it's an oxidizing environment, you're going to be producing sulfuric acid, which is a great acid for dissolving minerals. And if you don't have that pyrite, the rate at which the rest of the rock is breaking down will be a little bit slower as a consequence. And of course, the carbon implications of those different reaction pathways is going to be different as well. So yeah, reactivity matters a lot. And I would expect a really different solute flux coming out of a quartzite versus a shale versus a limestone terrains. All these are very different in the way that they're going to be producing solutes. Does that get at what you're asking?
Yes, thanks.
Right. Thank you, Stan. And yeah, so I think we are on time. Let's give another round of virtual applause to our speakers. It started with a simple proposal, but evolved into this large session with all these fantastic talks. And I think the last part will be Libby and David will give a session summary and wrap up our session. If you're here, please feel free to take over.
Sure. Again, I'm David Button. I apologize if there's noise in the background. My house is actually pretty busy at the moment. But so Libby and I, we've been taking notes. We're trying to kind of summarize everyone's thoughts into some cohesive package. Who knows if we got there, but hopefully everyone got a lot out of the talks. But I think one thing I want to stress is that this session was really designed to highlight the LOAC, right? The land ocean aquatic continuum, with the idea that, boundaries that have been used in the past that have been finite and compartmentalized, we're really moving beyond that. And we understand that these boundaries are not finite. And in fact, there's a lot of processing and dynamics that happen that require us to think about this as a continuous environment, continuous set of processes and systems. And I think the talks really emphasize that this is the case, and this is the paradigm that we're hopefully moving forward into. And I like the comment and the discussion around communication, because again, this is more emphasis on the science side, but our ability to work with terrestrial ecologists, terrestrial system scientists, into freshwater ecosystem ecologists, into estuarine and ocean systems is good practice, right? It's good practice for us to learn the terminology, the language, the acronyms across all of those domains. And that's, you know, we can use that to go into more management environments as well and sort of create a common lexicon. So I thought that was really interesting. And so just to reiterate, and Libby, please feel free to jump in. I've missed some key points. The idea was that we started with terrestrial systems and river networks, right? And Suzanne, although you weren't here, I think that it was really important that we started as far upstream as we could go, to the rocks, the weathering processes, and highlighting that time is very important. And time is really important in terms of the short-term processes and long-term processes of what gets broken down, and the hydrogeobiological processes that then give us and set up our ingredients for what's going to happen in the aquatic system. Nicole really brought that forward with trying to identify those sources and the reactivity, in particular of minerals, right? And in particular carbonate species with the minerals that are coming into our aquatic environments. And there's a number of different factors that are going to drive that, and it's different in any aquatic system as the science continues to show. And as the scientific community is emphasized, and if you went to AGU last year, this is why this field of enhanced mineral weathering has started taking off. We can sort of throw minerals on a field and understand those short-term and long-term mineral dissociation processes. Maybe we can actually evaluate that in the context of carbon drawdown. We shall see on that one. More importantly, well, not more importantly, but just as importantly, Jennifer really started to make us think about land cover change and land use change, and how that really imparts and it restricts the carbon composition and the processing of particulate organic matter export moving downstream. And Shuang, once we sort of understand what was coming into the system, Shuang was able to highlight from an organic perspective that the aquatic environment, the aquatic network, the freshwater network, it's not static. We've moved beyond the paradigm of thinking that it just shunts material out. And in the understanding de-ice dynamics, it goes in all directions. It comes in and out of the system. It moves across the air-water interface. And transformations of organic carbon to inorganic carbon along the network is really critical to understand what we're feeding out to the coastal environment. And there's a lot of work to be done, both from a modeling perspective and an observational perspective on the temporal and spatial dynamics related to that. Jennifer further really dove into highlighting uncertainties associated with organic matter transformations within the stream network, in particular photodegradation. Please don't want to put more words in people's mouth. This is my take. But right, this is where things get really tricky, because I don't think we, in the science has shown, we haven't necessarily devoted enough attention to the photo degradation microbial interactions and how those transformations play out spatially and temporally in these networks. And then Blake brought a project together really emphasizing, so if we're going to think about these land, ocean, aquatic network and think about it in the context of carbon and carbon sequestration on carbon credits, do we have the information we need to actually mass balance that out and figure out if this continuum is a critical piece that is not properly balanced? And if we're going to try and throw money at this from a credit perspective, perhaps we're not there yet. There's a lot of uncertainty. And Tom Bianchi did a great job summarizing a lot of the work, but I just want to emphasize from his perspective that we cannot forget about POC, the particulates that come out in all forms, right? And the importance of this space within the terrestrial environment, critical zone, that contributes all of these materials as it moves downstream. Some of the gaps that came out from that session, we still really need to understand, and this is actually a gap that I've made note of throughout each of the subsections, is that disturbances are still a major gap in our understanding. You know, these events, we know that these events are potentially going to increase in frequency, But what do we do when we have this pulse of energy through the entire system that can affect weathering rates, that can affect, you know, the sort of kinetics of respiration, photosynthesis, and what does this mean for the transformations of organic to inorganic carbon, and ultimately the processing that we shoved out into the coastal environment? So that wrapped up sort of the upstream component. We moved downstream, and we had speakers related to wetlands and estuaries, right? And so this is where we start to jump into where freshwater systems interface to saline environments. Kipe and Lisa Marie highlighted two systems on the East Coast and the West Coast that show the complexity of how complex these systems can become when we mix all of the upstream biogeochemical dynamics into a saline and tidal environment. But I think one of the things that came up from their talk is the emphasis that, you know, perhaps we really need to center this land ocean aquatic continuum and bring in the human component. And that's I think she mentioned something, the LOAHC as a new acronym. And this is a really key piece that sort of fits into a potential gap when we think about it in the context of disturbances as well. Etienne provided a summary on sort of the importance of wetland systems from lateral export, but again, stressing again, just how much disturbance and wetland loss has already occurred. And so allowing us to think more about how the past should help us inform the future, whether that's from a restoration perspective or for a carbon uptake perspective. And then Jeremy reminded us that it's not all about carbon. And I think this is sometimes a refresher for the OCB group, is that in order to understand the complexities of these carbon dynamics, and his work in the Chesapeake Bay really underscores the fact that there are a whole bunch of controlling factors, nutrient dynamics that are actually underpinning the processing and transformations of carbon as well, adding complexity to the models that we need to address. So some of the major questions that came out from that section, you know, do we have the information that we need to really understand the interaction between sea level rise and hydroclimatic variability? Again, thinking about if we have a pulse of energy and water coming through the terrestrial system, through the aquatic network, interacting with sea level rise, where is that interface changing? Is it moving upstream? Is it moving downstream? And what does that mean moving forward, right? And one of the big aspects that might be something that could be addressed is focusing on, and this kind of gets to John Kominoski's point, focusing on the water residence time, really understanding where water sits along this continuum, for how long, and how that relates to the chemical transformations that we're starting to get a better handle on. So that was kind of exciting. And then finally, the idea was, let's keep moving out, right? Let's go to the coastal environment, right? And keeping in line with this LOAC framework, right? That it's very hard to differentiate this boundary between wetlands, estuarine, and the coastal environment. And I think John gave a great overview this morning about what does that mean in the Everglades system, right? You have a system where you're seeing sea level rise, but also freshwater inputs increasing over time, and two different environments that are underlain by different sort of wetland ecosystems operating differently when it comes to carbon export, right? One, you're sort of seeing an inflow of oceanic biological carbon moving into the system, whereas another one where you're seeing more of a humic outflow or increase in humic carbon being a proportion of organic matter in the environment. And so there's a lot of work to be done in this sort of tidal hydrodynamic space. You know, Gloria, again, presented some of her findings related to the role of alkalinity in blue carbon systems, and in particular in mangrove systems, and, you know, shared some of the work that there's still a lot of gaps in our understanding whether these mangrove systems are in fact major carbon sources or sinks or net neutral, where DIC is still considered the largest outwelling, largest export component. But it's that sort of unaccounted for component that is still a bit of a mystery in there. But the potential for mitigation is large. And I think that's a really exciting area that research is showing we need to move forward. some of the tools that we have at our disposal right now, and some that are coming down the pike. Cedric was able to give us confidence that organic carbon and CDOM is actually measurable in sort of a robust way, and that we are having a new approach and evaluating approaches to use satellite remote sensing, hyperspectral data, high temporal resolution data, along with Joe Salisbury's update on Glimmer, fingers crossed, that we're going to have the temporal resolution to potentially really start to understand dynamics, disturbance events, pulse events that the human interactions may actually be showing. And from that human perspective, Xinyu and Simone were able to spend some time talking about anthropogenic carbon uptake, both on the East Coast and on the West Coast. And really, you know, again, coming from these two particular talks, I think what to sort of summarize some of the findings related to that is that we're actually starting to understand the spatial variability and the spatial dynamics of where anthropogenic carbon uptake is happening most efficiently, most effective, most intensely. And in some cases, it's in the nearshore environment. In some cases, it's in more of an offshore environment. And understanding where in space those processes are actually occurring has been pretty exciting. Whether that's good for Dungeness crabs, it sounds like the jury is still out. If we want a bunch of small crabs, maybe it's a good thing. Thanks for that, Simone. And so, Livy, I don't know if you want to add anything, but I think it's been really exciting to see this group of talks come together along this continuum, and hopefully we've been able to start to really evaluate this carbon cycling in a more continuous manner, and it's a good pathway to keep going moving forward. Libby, I'll let you add anything.
Yeah, I don't really have much to add. David did an amazing job summarizing everything, and I just want to put a plug in that that, you know, part of why I greatly appreciated being able to participate in the organization of this is that I am the coordinator for the North American Carbon Program, which was introduced on the first day as, and we have, you know, traditionally been more terrestrially focused, but we have been working with OCB over the past few years in really trying to support more integration across our communities and to really get at all of these interesting and very important questions that that have been presented here during this session. So I'm really glad that I was able to participate, and thank you so much to all of our speakers.
Thank you so much, David and Libby. That's a great summary. Shaily, Kangchang, do you have any other things to add, or can conclude the session?
I echo Libby's sentiments. I'm really happy to have been able to participate.
Yeah, I agree. That's
I
great.
agree.
Right. Thank you all. Thank you so much. I will hand it over to Heather if you have any announcements.
Thank you so much to all of you. Let's give our speakers one more round, and our session chairs, because this session was a bear to pull together. This one it was big, and I think that our changing view of the land-to-ocean aquatic continuum is a more continuous system, and it's not just a one way. It's you know, moving in 3 dimensions. It's incredible, and I really enjoyed working with this group